Hello everyone. My name is uh, Pedro Raposo, and I'm a curator at UniProt. And in this webinar, myself and David Carpentier, which is another curator at UniProt, we are going to talk about our automatic annotation systems within our database. So we are going to start by discussing how UniProt uses these systems to add annotation to a large number of uncharacterized protein entries. And I will start with a short introduction on UniProt, and then we will look into the annotation systems in depth. And afterwards, we will finish on how UniProt made it possible for you to annotate your own set of proteins using these tools. So to start with, UniProt is the world's leading high-quality and freely accessible resource on protein sequence and also functional information. We provide reliable data as we have teams of expert curators reviewing literature in order to describe the function of different proteins. And we are a consortium composed of 3 different teams. We have MLBI from the UK, the Protein Information Resource, or PIR, from Washington, D.C., and also the Swiss Institute of Bioinformatics, or SIB, in Switzerland. And our efforts coalesce together to provide you many of the services that we have on our website. Then, when we enter into the website, we are welcomed by this homepage that you can see here, where you can find a central search bar to find any specific protein or group of proteins by inserting a more general term. At the very top, you will find some tools that will help analyze your proteins of interest. And the resources that you have in UniProt are diverse and serve different purposes. Here we have here a diagram on the interaction between all of these resources. So as a short introduction, we start by importing information from external genomics resources, such as DNA or GenBank, for example. And then we archive these protein sequences in UniPark. So UniPark is our archive sequence resource. And then we transform these sequences into protein accessions inside UniProt knowledge base, or UniProt KB. And this is where they get the annotations. Also, we can organize these protein accessions by their organism, and therefore we create proteomes. And finally, we can organize these proteins depending on their similarity, and we cluster them using UniRef. But today we are mainly focusing on UniProt KB, which is our most used resource. So the knowledge base is composed of 2 different data sets. We have SwissProt and Tremble. SwissProt are the records that contain reviewed information, which means that a UniProt curator has read through the literature with experimental data about this protein and added to that record. And you can identify these with this icon with the golden star. And at the moment, we have about 500,000 proteins from SwissProt. And then we have Tremble, which is the data set of our unreviewed proteins. And we have around 200 million proteins of these at the moment. And these have annotations from our automatic annotation systems. So the types of information we provide in UniProt are quite diverse. For example, we have a protein name. We can describe the function using a comment, the taxonomy of the organism in which the protein actually comes from, where the protein can be located subcellularly, and its 3D structures from AlphaFold, for example, domains and motifs, several cross-references, etc. We know that it would be great to have all the proteins that we have within UniProt knowledge base to be manually reviewed, but however, this is not possible because not all of them have experimental data. So what we are tackling this issue using automatic systems. Another issue that we have is the growing number of imported proteins coming into UniProt. So there's a rapid increase due to the advancements in sequencing technology, and these allow scientists to sequence genomes faster and with better quality. So this results in genomes and proteins, more and more proteins being available to us. And taking this into account, manual creation is just not enough to characterize all proteins. And so to address this, we are using our automatic tools. So initially, UniProt creates, when UniProt creates a new protein accession, it starts by definition to come as an unreviewed entry. So it will be part of Tremble. And if that protein has some experimental data, then it is possible for curators to curate it, transforming it into a SwissProt entry. On the opposite side, SwissProt entries are used as templates to create rules to automatically annotate, to propagate annotations to Tremble. So as a summary, our goal with these systems is to provide functional information to proteins that cannot be manually reviewed. And these systems are reliable and traceable, as we are going to see in the next slides. So in order to propagate annotation automatically, we can start by thinking UniProt knowledge base as a space where many different types of proteins exist, and the space cover a variety of different proteins coming from different organisms. So firstly, we can group these proteins by how similar they are. And then what we observe is that inside of these groups, we have a mix of proteins some of them have been reviewed and some of them are unreviewed, and they are color-coded with orange and blue, respectively. And we can think of these a bit more abstractly. We can say that each group here can be composed of a SwissProt space and a Tremble space. So after confirming that each group has homogeneous proteins, and by that I mean that all proteins from a group have the same annotation, we can propagate information from SwissProt to the Tremble space. And this is because all proteins from the same group are very similar. And because SwissProt are the manually reviewed proteins, then we can infer that the Tremble would have the same or similar annotations. So each time we infer information this way, we say that we have a rule for this propagation. And finally, we do not consider groups that we do not have any experimental evidences, so we cannot infer anything in these cases. So this is another way of visualizing what we just saw. In the first example, in group 1, we have to group these proteins together as they are very similar. If we know the function and the characteristics of the orange proteins, which are SwissProt, because they have been manually reviewed by curators, we can infer that the unreviewed Tremble blue proteins have the same function. Now, the types of annotation we can propagate will depend on originally was curated from the SwissProt proteins. So in the first example, we can see that we can propagate the name and the EC number. But on the second group, maybe we can just propagate the function comment and subcell allocation. So this is an example of a rule in UniProt. We can see that it's divided into two parts, and the left part is the conditions, and the right part is the propagations. So the conditions describe how to group these proteins together, what they actually have in common. In this case, we group proteins that have these PFAM signature that we can see here, and that come from eukaryotic organisms. And we are going to see later what signatures actually are in the next slides. And at the end, any tremble proteins with these two conditions will automatically get the annotations described on the right panel. So in this example, they are going to be automatically given this protein name, this subcell location, and all the other annotations that you can see here. So just to show you how this looks like in the end, we have a tremble entry, and you can see this protein comes from a human, and it has the function comment required for normal Golgi function. And you can see that this comment was propagated by our automatic annotation systems by looking at the tag attached at the end. So if you click on it, you can see the types of annotations and the specific systems that provided this annotation to this protein. This is how we make our annotations traceable. You can see this was provided by both UniRule and ARBA, which are two different systems that we are going to discuss. And where UniRule is a more controlled version of automatic annotation system that is controlled by curators, where ARBA is completely fully automated. And both of these work together to provide the best annotation for each protein. When it comes to the UniRules themselves, these can be created by different teams. So as a reminder, we are consortium. So depending if it's created at EBI, PIR, or SIB, you can have different IDs here described at the bottom of this page. But all of them come under this unified ID, this umbrella on UniRule starting with UR. So you can search by this unified ID our rules. Now that we covered what a rule is, I'm going to explain a bit more in detail how these are created and maintained. So we start by looking at our SwissProt space. This is a reviewed entry, and you can tell by the golden icon at the top this is a SwissProt entry. And if you go to the family and domain section, you can see that the PFAM that we used in the previous rule shown to you. So what are signatures and why are we using it in our rules? So simply put it, a signature is a hidden Markov model or an HMM, which are basically probabilistic models that describe functionally a region of a protein. Now, an HMM can be very small in length and describe the domain of a protein, or large enough to describe the function holistically of the whole protein. And the purpose of using signatures in these rules is to group them together. So we can infer information from SwissProt to Tremble. Now, these signatures are found and imported from Interpro. And Interpro is a database that conglomerates information from about 15 different data sets with data on functional classification of the signatures. And there are a number of signature databases that try to infer a protein function in very different ways by using different methodologies. But Interpro comes together and brings all of those, and we can import our signatures from this unified database. And this is an example on how Interpro displays signatures from a protein. So each color bar represents a different signature here. And on the top horizontal gray scale, this represents the protein that has about 800 amino acids. And we can see on the right the signatures that this protein contains. And here we have marked the pfem signature that we used for the rule previously shown to you. Yeah. So let's see how we can create a rule using the pfem signature we just looked at. So first we want to group proteins together using it, and we need to understand what are these. If you go to the advanced search, and you can search for these pfem specifically, and here in each field this is customizable, so you can customize to search for pfem only. So if you go here and select pfem, then you can introduce the ID that you want to search for. And also we were looking into organisms coming from eukaryote only. So with these two conditions, you can search, and this will result in 3,239 different proteins. And you can see that this set it's good enough to use to create a rule because we have a set of SwissProt and a set of Tremble. So what we want to understand is to see if these 35 entries are consistent enough in their annotation to see if it's possible to infer information to the other 3,000 Tremble entries. In here, from the 35 SwissProt, we provide 3 examples and some of their annotation. So you can see that each row represents a different protein, and we can see that the similarity comment in the 6th field, it's common between all these proteins, and it says belongs to COX6 family. This is consistent enough. And if it's consistent between these 35 entries, then we can assume that the signature is accurate, and we can provide this into a rule because it's consistent between all of them. And the same thing can be said on the subcell location. All of these annotations from the different SwissProt coming from different organisms still have the same annotation. However, if we look into the function, we can see that the function of these 2 organisms, which are fungi, have a more specific annotation compared to the non-fungi, which in this case is human. So there are still some differences, and this is why manipulation is required to understand biologically how much specific we can be when creating these rules. So at the end, what we have, yes. So at the end, what we have is a rule which can be can be broad enough to propagate all these annotations from such as protein name, subcell location, similarity, subunit, keywords, and gene ontology terms. But then when it comes to the function comment, as we've previously seen, this function is depending on the source of the taxonomy. So we are going to create what we call case. In the first case, if the protein comes from a fungi, then it will have a more specific and a more tailored function comment. But if it's not coming from a fungi, like we've seen with the human, then it's going to have a more general comment. So this is how we kind of tune the granularity of the protein of the rule for different types of proteins. So these are the types of annotation we can add to the rule, and these ranges from the function comments, function pathway, gene ontology terms, you see numbers, etc. We have many. And all of these enables for you to have a clear picture of what is the function of the protein, but also to provide some context around it. And we are able to add annotation from the sequence as special conditions. There are 2 different types of annotation being added here. So firstly, bioinformatics software for transmembrane regions and signal peptide predictions is run on each of these sequences. And this particular protein has some evidence of transmembrane regions. So that would be part of the annotation within the rule. The system that I've been describing so far is called UniRule, and where the rules are created manually by curators. We have another automatic annotation system that is fully automated. It is important to note that these do not operate in the same way, but they complement themselves to provide the best annotation for each protein. So this system that we are presenting now, it's called Association Rule-Based Annotator, or ARBA for short, and it also uses the manual annotated set from SwissProt as the training set. So first it loads the data and then it filters in order to create a highly reliable data set for training. And in the next step, it will create the rules and select the most reliable one. And it aggregates them in order to remove redundancy between these rules. And it also performs a cross-validation in order to ensure the qualities through many statistical analysis. And then it outputs these as a report. And in the end, it propagates predictions that go into Tremble, just like UniRule. So ARBA is called a multiple class predictor system. And simply it means that it generates a model for all annotations we have previously mentioned. ARBA collects an exhaustive knowledge that is available to all data and selects only the rules that satisfy the statistical confidence criteria, which is 95%. These rules are then subsequently filtered down to a set of representative rules so that we don't have redundancy. So this means that we are filtering out rules to predict the same set of proteins with the same annotations. And now that we cover individually how UniRule and ARBA works, let's go through some differences and how these complement each other. So ARBA generates one annotation for each rule, whilst UniRule contains many different annotations for each rule. Another difference is that one protein can have multiple annotations coming from multiple ARBA rules, but it can only be touched by one UniRule. And at the moment, the difference in numbers is ARBA we have around 81,000, and UniRule we have around 9.5 thousand. So in the UniRule rule previously shown, you can see that we have many annotations within just one rule, but in ARBA, we provide here two examples, and in each one, you can only see one type of annotation. Additionally, UniRule and ARBA provides sequence feature annotations. So they annotate certain regions of the protein sequence. For example, they can flag where a certain domain is predicted. Also, we are adding the results of the sequence analysis methods, or SAMs for short, and these predict disordered regions of the protein sequence. They predict transmembrane regions, signal peptides, and coiled regions. So the sequence annotation covers half of the Tremble so far. Another system we have introduced is the Protein Natural Language Model, or ProtNLM, developed by Google. We have a collaboration with Google on the validation of this tool as well. And ProtNLM uses a transformer model, which is trained on the protein sequence alone to predict its name. And at the moment, it uses an ensemble of 7 models to predict the name. 3 that analyze the protein sequence, 3 that analyze the organism taxonomy, and an additional one if we have an AlphaFold structure. So to put it simply, each model provides a score individually, which is calculated at the end to have a final decision on the name. This diagram represents the number of proteins in the UniProt knowledge base. So the large green dark circle represents Tremble with about 202 million proteins, while the red circle represents SwissProt with 574,000 proteins. So overall, UniRule and ARBA covers about 61% of Tremble. And if we take into account all of our automatic annotation systems, we can cover 89% of Tremble so far. And to finish our presentation, we will talk about how you can annotate your own set of proteins locally in your computer. So we know that functional annotations that the systems we are giving are very valuable to you and for the scientific community. So we are sharing these using rules so that you can use these rules. So in order to share these rules, we have created the file format in which we can represent UniRule and ARBA rules. We have called it the UniProt rule markup language. It is based on well-supported and understood standards like XML, and it exposes the logic of these rules explicitly. So to allow the users to use these rules, we have created and released Unifier as an open source software. And Unifier is the tool that is going to annotate your proteins. It is described here on the right in this workflow. And you can see that the inputs would be the protein sequence, the taxonomy data associated with the protein sequence, and the signature data provided by Interpro. Then the rule engine using UniRule and ARBA rules would predict the annotations for each protein. Now, the Unifier requires several tools and dependencies to be installed, but you don't need to do any of this. You don't need to install it manually because we provide Unifier as a container, which is, as you can think it as a folder with all the software required to run. You just need a container software such as Docker Singularity or Podman. So you just need to install one of these and then have a FASTA file with the proteins and taxonomic information. And this is what we show here. So in order to annotate your proteins, this would be your input file. And this is a protein multi-FASTA file where each header is in the UniProt format. It needs to contain at least two types of data: the protein identifier and a taxonomy identifier from which the sequence is originally from. Now, on this tax ID, it's important to note that the more specific you can be, the better the annotation is. And what I mean by this is if you want to annotate a protein coming from a human, it will be best for you to add a taxonomy of the human and not the taxonomy of mammal or eukaryote. So in this slide, it shows all the steps that you need to do to run Unifier on your environment. First, you need to check if Docker is installed or Singularity. And afterwards, you can clone our publicly available GitHub repository of Unifier. And it comes with a script that runs everything you'll need. And this script provides a couple of options, but most of the options, most of these options have good default values, so you don't need to touch this unless you go for a more in-depth analysis. All you need to give to the command line is the script with the input file and the output directory. And with this said, you can simply run very quickly and easily Unifier to provide all the annotations that we could provide as a service coming from UniRule or ARBA with your own standards at your own time and locally in your computer. Just to finish the presentation, I'm going to mention some of the major changes that are going to come to fruition in the next UniProt releases. UniProt is going through some major changes in which some of the proteins from the UniProt knowledge base are going to be moved to UniPark. Now, some of these annotations would be transferred to UniPark, and some of them would be started to be calculated using unifier within UniPark. And I will show an example. So this is a UniPark entry, which is a bit different from what it used to be in the past. So we updated these recently to appear a bit more like a UniProt knowledge base entry. And as you can see here, the predictions have been generated using a unifier from UniProt, and they are reflected in this case in the function comment, catalytic activity, and this active site. As you can see, these have been manually or automatically annotated. If you have any questions or feedback on our automatic annotations tools, if you want to understand a bit more how it works, or you want to report something that you've seen in a UniRule or a ARBA rule that doesn't seem quite accurate, you can also contact us through our contact page. And if you want to read a bit more about UniProt, we have this latest publication in 2025. And more specifically, we have a different paper on UniRule itself for more details. And also a preprint on ProtNLM as well. So I want to thank our three teams and our funding as well, our funders. And just before I finish, we have several MBLBI webinars that are happening throughout the year. If you want to know a bit more information, you can visit our website on these events. And when it comes to UniProt, we are going to have on the 16th of April a webinar on antimicrobial resistance and its curation with David. And with this, I thank you, and we are going to have some questions now.
Great. Thank you very much, Pedro, for the presentation today. Now the floor is open for questions. There's a comment in chat about internship in EMBL. So EMBL has a job web page. So if you go there, they announce all sorts of openings which are available across the EMBL site. So EMBL has 6 sites. So you will see any kind of opening, including internships, advertised there. So that would be the best page to go to. You can type in more questions in the Q&A box. I think we have a new question. An automatic annotation I often see is tree grafter. How is the quality of this annotation checked for tremble entries?
I don't have much experience with tree grafter. I don't think this is an automatic annotation. This will be a cross-reference, I think. So we cross-reference out to several external resources. I'd have to take a look at this a bit more and get back to you.
One question that came in the chat, how we can specifically go to bryophytes specific annotation?
So I'm not familiar with this bryophyte. I assume this is an organism. If that's the case, then if you want to search the rules that would affect bryophytes, if you want to specifically look into the rules, you can go to our website, and in the main search, you can select ARBA or UniRule, and you can select by these word alone. If you want to get the annotations from that are touching all bryophyte proteins, then you need to search the proteins themselves, download and get the annotations from ARBA and UniRule.